And that's a good opportunity for me to take one moment of appreciation. This event was organized by a number of participants, but there's one key person without whom none of this would have happened, and that is Giselle Grenier. I love embarrassing Giselle, so thank you so much for everything you've done. We still have a half day ahead of us, but everything, the logistics, getting everyone here. Yep. Yeah.
Registration, thanks.
Yep.
Food, thanks. Everything's thanks.
Right. All right, so I know that I stand between you and some very, very impressive music, but I wanted to take a moment to tell you why we are interested in music, apart from avid consumers and enjoyers of this human activity, and tell you a little bit about animal work that's been relevant, I think, to what you've heard this morning already. As I said, the Conte Center is interested in neuroscience of mental health, mental illness, and our center in particular focuses on development as the origin of most, if not all, mental illnesses. And this comes from centuries, actually, of insight, but a lot of knowledge from the past 100 years in particular. And since we have a strong Italian component to this event, I would like to start with this diagram in one slide to tell you why we care about development. Maria Montessori was one of the first MDs, female MDs in Italy at the turn of the last century, and is very well known to all of you from the schools, honoring her great insights and pedagogy. She, through careful observation as a pediatrician of child behavior, came up with what she called the bulb model of behavioral development, long before we knew anything about the brain or critical periods or how this sensitivity waxes and wanes. And many of these elements are quite prescient. So she noted that a lot of behavior is even shaped before birth, before age 0. But then the first 3 to 6 years are a particularly important period of time when the environment can shape infant behavior. And of course, school starts at age 6. So all of this is happening before any kind of formal schooling. And then perhaps with adolescence, puberty, there's a second opportunity to shape other brain functions. And so fast forward to today, the work of our center, as well as several other laboratories around the world, have identified the biological bases for these windows. And perhaps more importantly, we now know that these windows of opportunity for shaping brain and behavior are also windows of vulnerability to mental illnesses which emerge at different time points in this trajectory. And so by understanding the mechanisms of how these windows come about, we hope to develop interventions or corrective measures as needed for these kinds of conditions. And so the work of the center and our lab in particular has focused on molecules and circuits in the brain that trigger or open these windows, determining the timing of these critical periods, as well as mechanisms that close these windows. Surprisingly, we now know it's not just a passive decay or loss of what we call brain plasticity, but perhaps even an active closure with brake-like factors that are expressed throughout life to make sure that the brain does not keep rewiring too much. And that opens opportunities for lifting the brakes in therapeutic settings. And so in 2 minutes, I'd like to give you a sense of what's going on during this kind of window of brain plasticity using a video that we put together at the Harvard Center on the Developing Child, which captures quite nicely, I think, this age-old discussion of nature, genes, and nurture, environment. So hope you can hear this.
A child's experiences during the earliest years of life have a lasting impact on the architecture of the developing brain. Genes provide the basic blueprint, but experiences shape the process that determines whether a child's brain will provide a strong or weak foundation for all future learning, behavior, and health. During this important period of brain development, billions of brain cells called neurons send electrical signals to communicate with each other. These connections form circuits that become the basic foundation of brain architecture. Circuits and connections proliferate at a rapid pace and are reinforced through repeated use. Our experiences and environment dictate which circuits and connections get more use. Connections that are used more grow stronger and more permanent. Meanwhile, connections that are used less fade away through a normal process called pruning. Well-used circuits create lightning-fast pathways for neural signals to travel across regions of the brain. Simple circuits form first, providing a foundation for more complex circuits to build on later. Through this process, neurons form strong circuits and connections for emotions, motor skills, behavioral control, logic, language, and memory during the early critical period of development. With repeated use, these circuits become more efficient and connect to other areas of the brain more rapidly. While they originate in specific areas of the brain, the circuits are interconnected. You can't have one type of skill without the others to support it. Like building a house, everything is connected, and what comes first forms a foundation for all that comes later.
I hope that gives you a sense of the amazing work that's going on in every infant's head in those first few years of life, and the enormous task of assembling a brain. This, of course, was a schematized version of what's going on, and some of the state-of-the-art science that's happening right here in this building is making those diagrams something much more real. This is work by Jeff Lichtman's laboratory here as part of the Conte Center, where they've developed, I'd say, medium throughput connectomics at the highest possible resolution by automating all of the labor-intensive procedures of electron microscopy and reconstruction of neural circuits. Imagine taking very, very thin nanometer-sized sections through a piece of brain tissue and aligning them one slice after another to reconstruct painstakingly every single element that's found in the brain. And to do this for the entire brain would take a very long time, but Jeff's lab and others around the world are approaching this systematically, and it's becoming reasonable to think of mapping small circuits in actuality. So in fact, if you look at a section of the brain like this, you'll see that there aren't any gaps at all, even though that cartoon showed a very sparse neural network. And you'll see elements of presynaptic and postsynaptic connections, and you can assemble what's called a connectome. And here's a small piece of a dendrite, the receiving end of a neuron in gray, with all these little spine-like protuberances receiving colored inputs called axons. And we'll fly through this very short segment of a dendrite from one of these reconstructions from a mouse brain, and you'll see that it's quite complex. But this is the real circuitry in the brain now, and it's possible to start imagining how we would map and visualize circuit changes in normal development during critical periods, what happens when you change the environment, how do these circuits rewire, what happens if you disrupt genes that are linked to mental illnesses, and so forth. So it's a very exciting time to be in neuroscience because the cellular tools are really unprecedented. And the question for us here today is, how can we apply this knowledge about triggers and breaks and circuit mapping and then functional and behavioral readouts to the question of what music is for, as Professor Zatori started us out with this morning. So music is wonderfully multimodal, very rich. It evokes both motion and emotion. And you can see from this simple cartoon that it has so many whole brain influences that we all enjoy about music. And so I thought I'd take a few moments in this talk to illustrate what you might be able to do with animal models, because the question was raised whether or not they are perceiving the music or feeling the music like we do. Of course, we wouldn't really know the answer to that question, but we can try to make inroads in some sense by doing experiments. So while Charlie Brown is probably experiencing the music like we would, dancing with joy and a happy smile, whether Snoopy is also feeling all of that is an open question. And so Psyche already mentioned the possibility that the animals can perceive the rhythms. And just at the end of last year, some colleagues at the University of Tokyo published a paper saying that rats can feel the beat. And so I'll show you this little video. So by using multi-point tracking, they claim that the rats here are bobbing their heads to the rhythm, in this case a classical piece. Right? And if you're not really convinced by that, then of course there's the famous cockatoo. This is not, this is not Snowball. It's another one off YouTube. It could be quite loud, so be prepared.
Gangnam Style. Gangnam Style. Gangnam Style. Gangnam Style. Gangnam Style.
This goes on for a while, so I think I'm going to stop. I would bet he's enjoying that piece, but I want to move from motion to emotion for this talk. So how would you measure some kind of emotional value or contribution of music? Assuming that our mice can also hear the music in that way, we, in this case, Eunjin Yang, a postdoc in the lab, devised an assay to try and determine whether they will develop a music preference by exposure earlier in life. And to do this, she borrowed what's commonly called the open field assay. So this is a top-down view of an open arena which mice can explore. And in the corners, we have little shelters. They're little plastic huts with nesting material, and in her case, little iPod headphones that can play music, which don't actually exit the doorway here, so the mice can only hear it when they're in the huts. And what you can do is track the motion of the animals. And what you find is that if you put mice in a new arena like this, they will initially explore quite a bit. But you can tell some features. They will go in and out of these huts in the corner, and they're generally fearful of the open center, which is a high anxiety location. It's quite bright, perhaps exposed to predators, and so they tend to go around the edges of these boxes. So here are two examples of that. But Eunjin was very patient, and she waited for several hours to see what would happen as the arena became more familiar to the animal. You can start to quantify how much time they spend in the vicinity of the silent chamber, if there was no music playing, versus a chamber playing music. And you see on average here, there's a lot of movement in the center. They're not clustering in one or the other chamber, but maybe biased slightly towards the silent side. But after 3 hours, it turns out that they make a choice. And that, in fact, in this case, the animal spends the last 30 minutes perhaps sleeping in one chamber, which happened to be the music chamber, and becomes a dot over here on the x-axis. And this one roamed around a little bit, but spent much more time in the silent chamber. And so in this way, we can assess two things: their anxiety level in the first 30 minutes, even though they have access to shelters, and their preference ultimately after 3 hours of exploration. And so using this paradigm, Unjin raised mice listening passively to music at different times in development. And to make a long story short, comparing 10 days of music exposure at postnatal day 15, so 15 days after birth, versus 2 months after birth, the same 10-day music exposure had very different consequences. So in this 3-part graph, you'll see that mice raised without any kind of music experience are generally neophobic, and they prefer to end up in the silent chamber 75% of the time. And that's also true of mice exposed to music later in life at 2 months of age. But in this group here, they're all tested, by the way, at 3 months of age, roughly. You see that the early 10 days of exposure has biased their preference against their natural tendency to be in a silent shelter and spend much more time in the music playing shelter. And in this paper, we did a number of control experiments. So if you had old music, familiar music versus new music, they prefer the familiar music. If you use something that's not really music, like tones, then they don't develop this bias, even if you use the same critical period window. And so we felt that this was perhaps getting at something that approaches what we call preference behavior in humans for music. We don't know what the mice are making of this sound that they never would normally hear. But having translated this to a mouse, you can take advantage of all the tools that we've used over the years in mapping critical periods, such as animals that have open-ended critical periods because we've removed some break-like factor. And the idea was, could we, in fact, introduce an auditory preference later in life in such mice? One example of this, and there are several breaks now that we are aware of, is a very coarse manipulation, and that is accessing gene transcription. So genes are encoded by DNA, but they are also made more or less accessible by what we call epigenetic manipulations. And so, in fact, the DNA is tightly wound up in our cell's nuclei, and certain enzymes make sure that it remains tightly wound, especially in adulthood. And so you can inhibit those enzymes and unwind the DNA, even in adult animals. And this manipulation actually seems to promote plasticity in adult mice, adult rats, adult humans even. And so what happens here? This is a different way to plot the same kind of data. Basically, we're showing the percentage of mice that prefer silence. And in the center, you see 3 curves all lined up. These are all adult animals before they're exposed to any kind of music. They have this natural preference towards silence. And if you treat adult animals with saline, basically salt water, you don't change their preference with music exposure. They're adults. But the manipulation of opening up their chromatin with the drug called valproate actually allows the music now to shift their bias away from silence towards music. And a different condition, removing a gene that acts as a brake, has the same effect. It allows music to shift their preference even in adulthood, or go the other way. If you keep them in total silence, then they shift even more in favor of silence. So the kinds of rules that we had learned in, say, primary visual cortex or primary auditory cortex seem also to be involved here in this kind of complex preference behavior. And we noticed something that took us into the brain and which circuits to focus on. And that was the first 30 minutes. If these mice that had been raised with music were put in the open arena, and they had one chamber with music, one with silence, you see, I hope soon, that these animals are a little more willing to cross the center. And so in the first 30-minute period, the center crossing time increases. I think soon you'll see it just goes back and forth across the middle between these two chambers, which is rather unusual for mice. Instead, mice that don't have any kind of music experience are doing their normal thing. They may cross the center once or twice, but generally will hug the sides, called thigmotaxis. So you'll see the behavior has been altered by the animal's past experience with music and the ability to dose themselves with the music now, months later, as adults. So it suggested we should look in a place that's related to anxiety and control of anxiety. And in fact, only in conditions where a critical period was open, like young mice, or these animals in adulthood with the brakes removed, do you see the center crossing increase significantly in the first 30 minutes. And the place to look in the brain for this turned out to be the medial prefrontal cortex, which is part of that reward emotion regulation circuitry that was mentioned earlier. So in a narrow sliver here on the left is the auditory cortex. In green are cells that are activated by listening to the music. And not surprisingly, the auditory cortex lights up above baseline no sound stimulation in both the plasticity unopened and plasticity opened conditions. In the medial prefrontal cortex, though, it discriminates. So this part of the brain is more silent, and as you see here, without any stimulation. And then when you play the music of their youth, then the prefrontal cortex lights up very nicely when the plastic window is open. In this case, it's adult animals that gained a preference, as opposed to unopened plasticity. And it's specific to music. If you use the same amount of 10 days of just passive tone rearing, you don't get that kind of preferential activation here. And so you can quantify all of that, and you'll find that, in fact, when the brain is acquiring a preference, say in these brake-removed adults or younger animals, you get engagement of the prefrontal cortex. So this was rather intriguing. Even the lowly mouse who doesn't produce music, doesn't usually hear music, seems to develop some kind of preference behavior for this stimulus. And so we wondered if we could translate this to humans so that we could identify a similar critical period for the anxiolytic effects. And for this, you need to roughly correlate the age of development in mice to humans. And there are a number of metrics, and this is a difficult comparison to make, but the critical period that we talked about is right here, 15 to 25 days after birth, which is roughly middle childhood in humans, before puberty and adolescence. And so we predicted that if there were to be a critical period for music preference that is anxiolytic in humans, it would be in those middle childhood years. So how would you translate something like this into a human study? Fortunately, around the time we published this work, we had a small conference, which speaks to the value of these kind of in-person meetings, where I had several dinners with Nim Tottenham, who was a professor at UCLA at the time, and she studies human brain development. And one of her PhD students, Laurel Gabbard-Durham, actually was a student in my class at Harvard as an undergraduate, had been studying prefrontal amygdala connectivity. And so we came up with a human version of this study. So around the time we published this paper, it was 2012. For a 22-year-old adult in 2012, 7 years of age, this middle childhood would have been sometime in 1997. And so is there some way you could use that knowledge to have a quantitative exposure to music that you could borrow and test now their preference as young adults? And there is. It's called the Billboard Top 100. And so at least back in the day when music wasn't available all the time everywhere, you could devise a study where you could use the metrics of airplay on the radio as well as the ranking and relative exposure that people might have had. And Laurel went to work, and she started producing clips of music for people who would be adults that might have been exposed in the United States growing up here to pop music, trying to match that to more recent songs in the Billboard Top 100 for all of these parameters. Just to give you a flavor of this is a kind of childhood song.
I can be your hero, baby. I can kiss away the pain.
And this is a more recent one.
There'll be no sunlight if I lose you, baby. There'll be no clear skies.
So we even matched the word baby in the so basically the idea was to take songs that didn't quite become global hits but got a fair amount of airplay had you been raised in the United States, and compare these young adults to immigrants to the Southern California, at that time UCLA area, who would not have had as much exposure to that music. And test whether there is a preference now in adulthood to adolescent music, childhood music, or even earlier music, what we call preschool music. And we were able to test a number of these subjects and find whether it has an anxiolytic effect by introducing a stressor. In the case of mice, we had the open field. In the case of humans, what would you do? So the idea was to create two simulated radio stations, one that was full of songs of more recent airplay, and another station that was full of songs from these different periods, and have the individual simply choose one of these stations to listen to before and after a stressful event. The stressful event we chose was a simulated math exam, like the SAT. And Laurel was quite devious. She made it even more stressful by putting them on a timer and telling them points were subtracted if they were wrong. And they gave them very difficult math problems with buzzers. When points were subtracted, she stood over their shoulder. She kept telling them how poorly they were doing compared to their peers. And the poor students who were doing this felt this was the test, but in fact what we were interested in was the music they chose after. And we repeated this cycle several times. And they had ratings of their stress level as well as physiological measures like galvanic skin response to corroborate. And I'm editing a lot of data here, but basically in the people who had exposure to that childhood music, compared to before the stress, they were going there more regularly after the stressful event compared to the immigrants who didn't have that exposure, who basically randomly or more often chose the more recent music during those timeout break periods. And this translated nicely to both self-report and physiological measures of calmness. So we felt we had a paradigm that was working, and in fact could probe what happens if you alter the critical period. We couldn't quite give drugs or knock out genes from humans, but one condition which sadly seems to hold up across animals and humans is early life adversity. And so children or animals that grow up in stressful environments seem to take a strategy of developing faster. And so here's an example of mouse work in the same prefrontal cortical areas that show markers of brain maturation coming up much earlier than normal compared to control care animals. And so we revisited the data set and pulled out those folks who had history of adverse childhood experiences, such as a divorce or stress in the family, that single parenting or poverty, a variety of conditions, and found that remarkably, those young adults were actually showing a greater preference for their preschool music a clear indication of a shift of this window as indicated here. This is ongoing work, but I thought I would share it with you because it nicely held up the hypothesis that there is a critical period for music's anxiolytic effects. A subset of these folks were then taken to the scanner, and lo and behold, the preferred childhood music was preferentially activating the prefrontal cortex as opposed to the adolescent songs, and the more it did so, the more calming it was in these people. And finally, a mediation analysis was done to look at the connectivity, functional connectivity, as you've heard before, between the prefrontal cortex and the amygdala. And it showed that there was a stronger functional connectivity between prefrontal cortex and amygdala that was mediating this anxiolysis. And so what I'd like to come out of this little presentation saying is that childhood music and music exposure and experience is beneficial in so many ways, from motor coordination to cognition. But here's one aspect that took us from the mouse to the human and suggested that middle childhood might be actually a critical period for this very important step of being able to control your emotion at this level. And so I'm going to stop here and thank the folks who are involved in this work. Eunjin Yang, a former postdoc, Eric Lin, an undergraduate who did a large part of that work as his thesis, was a musician in the Harvard Symphony, very interested in music therapy and ended up in medical school. Jean Leyer, a graduate student after them. And of course, Laurel Gabbard-Durham, who is now a colleague of Psyche's at Northeastern University, and Nim Tottenham, who has moved to Columbia in the meantime. So thank you very much. I guess I'm moderating this too, so if there are one or two questions, I'm happy to take them, but if Adam would like to set up in the meantime. Yep.
I wish they wouldn't use silence as a control in the animal studies.
Yep.
Because these are animals that are typically prey, not predators.
Right.
And music can mask out environmental sounds that can alert them to the presence of a predator. Right. So, a better, I think, control would be noise at the same level. Yeah. The other thing that wasn't clear to me was what kind of music were they giving to the rats or mice? Was it stepped up in frequency? Was it tonal? Was it rhythmic? That would be good to know.
Right. These are all great questions. So obviously, raising mice listening to music was a first for us, and so we tried to choose deliberately quite different pieces like classical and jazz, and just wanted to know if they would develop some kind of preference for what they grew up with. Your point about silence is well taken. In fact, there's never a silent moment in their lives, right? They're making their own noises. And this whole study was actually meant to be a toy model to test ultrasonic vocalizations which they make to each other and see if mice will develop preferences for who they were listening to growing up. And that whole study has been completed and actually fits this model very interestingly. Same critical period, but there's a clear sex difference there. So in those studies, we can jumble the syllables of the ultrasonic calls and have two kinds of similar sound stimuli, to your point exactly. But I did mention briefly that we used novel versus familiar music as one of the controls. It's not perfect, but there's a lot to do. So if there are any students interested in this kind of work, we have a lot to unpack. Yeah, thanks. Yes?
Thank you. Thank you, Takao. That was great. And I was really fascinated by the idea of the early exposure having this sort of anxiolytic effect. And I wonder what your view would be about the reasonably well-established sort of preference for music during adolescence that people like Carol Krumhansel have shown. And would it make sense to think that that has maybe more to do with social bonding and peer relationships versus the more kind of security aspect that's associated with early childhood? Are there two critical periods for different aspects of preference?
Yeah, thanks so much. So within any given modality, there are often multiple critical periods. And so I would agree completely with your suggestion. I think if you ask someone on the street, what music do you prefer, it tends to be from adolescence, but that's associated with positive social reinforcement and going to the rock concert with friends and so on.
And dopaminergic surge
Exactly.
at that time.
Yeah, so to pull out this one, I think we were lucky that we used the open field, which is an inherently stressful environment for the mice. And that's why we went to all that trouble with the math exam and trying to create a situation that was not a kind of positive reinforcer. And then you see the value of the music from the middle childhood coming in.
Yeah, it's wonderful. Thank you.
Thanks.